Thank you, everybody, for tuning in. So I'm very excited to be here. My name is Alex Muir, and I'm super excited to tell you about the work that our group here at U Chicago has been doing to look at the different nutritional and metabolic barriers to tumor immunity that occur in pancreatic cancer. So to jump into it, I'm going to talk about the cancer immunity cycle, which probably doesn't need that much of introduction here for this particular symposium. But just to say that this is the way by which cancer cells release neoantigens, and these neoantigens can then be presented to T cells, and T cells can then recognize those antigens, infiltrate into tumors, recognize cancer cells that express those antigens and kill them, and lead to the continuation of this cycle. And obviously this pathway and cycle is impaired by many different cancers to enable their progression and growth. But I'm going to talk about the ways in particular that that we've been learning that this cycle has been impaired in pancreatic cancer. And this pathway and immunity cycle, of course, has been targeted by a number of different approaches to rejuvenate the cycle, to overcome these barriers, to lead to a lot of durable tumor regressions and treatment in a number of different malignancies. And of course, CTLA4 and PD-1 based therapies for targeting priming and activation and killing of cancer cells have proven incredibly useful therapeutic tools in a number of different malignancies. But unfortunately, in pancreatic cancer, as has been highlighted by Florencia McAllister in this really nice review down in the bottom right-hand corner, these current clinically used approaches for boosting and rejuvenating the anti-tumor immunity cycle have not really had very much efficacy in pancreatic ductal adenocarcinoma, or PDACs. And this has led to a lot of interest in searching in the field to understand why these current immunotherapeutic approaches don't work. And this has led kind of this question of a number of groups in the pancreatic cancer field trying to understand where this immunity cycle is being broken in pancreatic cancer. And if we can identify these critical barriers to the immunity cycle in pancreatic cancer, can we clinically intervene and improve anti-tumor immunity and immunotherapeutic outcomes in this disease? And so in the last few years, there's been a lot of real awesome, tremendous work that has identified a number of different places in which pancreatic cancers stall this immunity cycle. And so there's been great work from David DiNardo's lab on the left-hand side here, showing that there's a profound defect in dendritic cells and cancer antigen presentation in pancreatic tumors, and that can stall out this cycle. There's been great work from Shukun Zhang's lab showing how different cell types in pancreatic tumors can prevent infiltration and trafficking of T cells into the tumors, so the T cells never show up to the tumors to kill the cancer cells and control these tumors in the first place. And then there's also been great work from Al Kimmelman's lab and Ruchika Pereira's group that have highlighted how cancer cells can use autophagy to downregulate their MHC molecules and slow their signaling and slow their presentation of antigen to T cells, and thus kind of trick the immune system in this way as well. And so these are all awesome studies that have highlighted a number of different ways in which this pathway has been broken, and ways that we can intervene to improve immunity in pancreas cancer. But I want to talk about one study in particular that I think is really highlighting that even if we're able to kind of bypass this cycle completely, that there's still some barriers to anti-tumor immunity in pancreas cancer. And this comes from work from Sunil Hingorani's lab and Phil Greenberg's lab at Fred Hutch, where they asked, what happens, can we use adoptive T cell therapies to control pancreatic tumors? And a quick primer on adoptive T cell therapy. This is where T cells are isolated from autologous T cells from patients or from donor mice. And these T cells can then be transduced with T cell receptors or chimeric antigen receptors, which on the right here, the CARs are synthetic receptors that bind to surface antigens on cancer cells, or the transgenic TCR approach uses native TCRs that can recognize tumor antigen that's being presented by MHC. And then these T cells are stimulated and excited ex vivo, and then they're reinfused back into the patient where they can go to the tumors and target the cancer cells and hopefully eliminate them. And so these groups at the Hutch, what they did was they identified the tumor antigen, mesothelin, over here on the left in red, which is expressed in nearly all cancer cells and pannins, precursor lesions, primary PDA or PDAC tumors, as well as in metastases. And these groups developed a TCR that recognizes this TCR 1045 over here on the right, that recognizes this mesothelin when it's presented on MHC. And they also developed a TCR GAG, which is a dummy TCR that doesn't recognize antigen. And when you transfuse mice with these TCR engineered T cells into tumor-bearing mice, as you can see, they're able to infiltrate the tumor quite readily, much more so than the T cells, the dummy T cells. And these T cells are able to cause a significant amount of cancer cell apoptosis in these tumors. And so the T cells are able to kind of bypass a lot of these barriers to tumor immunity and the tumor immunity cycle that we talked about and go straight to the tumors and start acting on them. Unfortunately, these tumors don't lead to, actually quite excitingly, this kind of adoptive T cell therapy is able to extend the lifespan of these animals, as shown here in these survival curves, compared to tumor-bearing animals that just are getting the TCR control T cells infused. But unfortunately, there's not durable responses to these, even multiple infusions of these TCR-engineered T cells. All of the mice eventually do succumb to this disease and have disease progression. And so why aren't these T cells, even when many of them are being infused repeatedly, why aren't they able to eliminate and durably control these tumors? And so one piece of evidence for why this might be occurring, why there's this kind of limitation to this approach, is down here in the right. And basically, as soon as T cells hit the microenvironment to these pancreatic tumors, they become profoundly dysfunctional and they're not able to exhibit their effector functions. And so these are T cells that are isolated either from the spleen or from the pancreatic tumor microenvironment. And if you take these T cells, ex vivo restimulate them, if the T cells are coming from the healthy spleen, when you restimulate them, they're able to produce cytokines, TNF-alpha and interferon gamma, in response to this restimulation. In contrast, the T cells that are coming from the tumor are profoundly deficient in their ability to produce these effector cytokines in response to restimulation. And interestingly, it doesn't matter if you're a T cell that's recognizing the mesothelin antigen or if you're a T cell that's not recognizing any antigen at all. The microenvironment is suppressive to the T cells regardless of repeated antigens exposure. And so what we think this is telling us is, going back to our tumor immunity cycle here, that a really profound and important block to tumor immunity is coming from the tumor microenvironment. That if you're a T cell, just being in that microenvironment is going to block your ability to be an effector T cell and to produce those molecules that allow you to kill cancer cells. And this is occurring even without antigen stimulation. There's something about this microenvironment that's inhibiting T cell function. And so this really led our group to ask, what about the tumor microenvironment is inhibitory to T cell function? What is in the environment that's so suppressive to these anti-cancer immune cells? And so one possibility that really caught our attention is there's been a lot of work recently in the field that, in response to immunological signaling, so signaling through the T cell receptor, co-stimulatory pathways or other cytokines, that as this leads to profound metabolic reprogramming in these cells. And I don't want to dive into all the details of this diagram here, just to say that there's a lot of metabolic reprogramming that's happening kind of under the hood of these T cells in response to this stimulation. And that metabolic reprogramming that happens is super critical for the ability of those cells to gain those effector functions. And what this means is, we go over to the right of this diagram, because these metabolic changes are so critical to the acquisition of T cell effector functions, that means the local nutrient availability can actually be a really important regulator of the metabolism that happens downstream in these T cells, and thus can be a critical regulator of whether or not the T cells can obtain these effector functions. And so levels of different amino acids, such as leucine, glutamine, or carbon sources, such as glucose, as shown on these diagrams, as well as other nutrients, such as trace minerals, all of these things have been shown to be really important regulators of whether or not T cells are able to gain their effector functions even when they're seeing these kind of immunological cues. And so we thought that this kind of the kind of nutrient regulation of whether or not a T cell becomes effective or not could be potentially really important in pancreatic tumors, because one thing we know about pancreas tumors is, in contrast to healthy tissues, which have well-organized vasculature over here on the right, which leaves these tissues really well perfused, and in the parenchyma of these tissues, the nutrient availability is basically roughly in equilibrium with the circulation. In pancreas tumors, in contrast, they have really abnormal and poor vasculature. There's excellent work from Dave Tuvison and Ken Olive where we can estimate that about only 25% of the blood vessels in these tumors are actually able to transport anything at any given time. And so these tumors are really hypoperfused, and this could lead to really abnormal levels of nutrients in this microenvironment, which could feed back in the inability of these T cells to respond to immunological cues in these PDAC tumors. And so we wanted to ask precisely this question: could abnormal nutrient levels in the PDAC TME, tumor microenvironment, could this be impairing T cell function in this disease and contributing to this exhaustion and dysfunctional phenotype that we see? And so to ask this question, our group has developed techniques where we can isolate interstitial fluid. This is the local perfusate of the tumor microenvironment from different mouse models of pancreas cancer. And then we can, and we've developed quantitative metabolite profiling pipelines where we can measure not just relative levels of metabolites in these interstitial fluid samples, but we can measure the exact concentration of about 180 major vitamins, nutrients, and signaling molecules in these microenvironmental samples. What I'm showing here on the right is when we take a look at metabolite concentrations in an unsupervised analysis of plasma samples from tumor-bearing mice versus these microenvironmental interstitial fluid samples. You can see that the TIFF samples, tumor interstitial fluid samples, are clustering apart and away from the plasma samples. So there really is a unique nutrient milieu or metabolic milieu in these PDAC tumors. And so what are the individual metabolites that are driving this kind of global metabolic difference, abnormal difference in these pancreas tumors? And so what I'm showing here is a volcano plot of metabolites that are enriched in interstitial fluid from PDAC tumors on the right or depleted on the left. And each one of these dots is an individual metabolite. And there's a few things that I really want to highlight. And the first one is normally when we think about the tumor microenvironment, I think kind of our gut instinct is that it's going to be a nutrient desert, right? There's poor perfusion, so there's poor nutrient delivery. So mostly there's going to be metabolites that are depleted in the microenvironment. That's actually not what we see. There's actually more metabolites that are enriched in the interstitial fluid of these pancreas tumors. And this includes a lot of metabolites that could have really profound roles on cell physiology. There's neurotransmitters like GABA, amino acids like glutamate and glycine that are accumulating in the microenvironment, as well as molecules like phosphoethanolamine, a Kennedy pathway intermediate, which I'm going to talk about in a bit. So I think that's one really important thing is to conceptualize the microenvironment of tumors as not just being depleted of things, but also accumulating metabolites as well. There's a lot of metabolites that also surprised us, which don't really change between the plasma and the interstitial fluid. Our kind of picture going into this study was that cancer cells are going to be the primary controller of what nutrients are in the tumor microenvironment. And so the things that they consume or release are going to be the things that are the most depleted or enriched in the tumor microenvironment. So things like glucose, lactate, and glutamine, these would be the things that would be most depleted or enriched in the microenvironment. This is not actually what we see. We don't really see a significant decrease in glutamine in these PDAC tumors. There is a decrease in glucose, but it's not amongst the most depleted metabolites, and we don't see an increase in lactate as well. And so we really are starting to wonder if our kind of hypothesis that cancer cells are controlling the nutrient microenvironment of tumors, if perhaps that might not be entirely true in every tumor type. But there are some metabolites that are strikingly depleted from the interstitial environment of these PDAC tumors, and this includes a number of amino acids such as arginine, cysteine, and tryptophan. And these are all amino acids that we know suppressive myeloid cells work to deplete from tumors. And so that's one thing we're starting to consider, is that perhaps the metabolic milieu of these tumors is not only responding to what the cancer cells are doing, but is really heavily influenced by what infiltrating myeloid cells are metabolically doing. And we also do see some metabolites like pyruvate, which is really critical for cells to adapt to hypoxic states, also being depleted in the microenvironment. These tumors are quite hypoxic. And so we're also starting to consider the hypothesis that perhaps tumor hypoxia is an important component driving some of these metabolic changes in the tumor microenvironment. All right, so we have kind of a parts list now. We know what metabolites are enriched or depleted in the tumor microenvironment, and we know what concentrations there are. How can we ask if these metabolic abnormalities in these PDAC tumors are contributing to the T cell function that we see in vivo? And so to ask this question, what we've done is we've took our quantitative measurements of nutrients in the tumor microenvironment, and we use this to develop a cell culture formulation that we call TIFM for tumor interstitial fluid medium. And this contains 115 polar metabolites at the exact concentrations that we measured in these microenvironmental isolates. And to make the complete media, we add these metabolites to dialyzed FBS to provide them to provide cells the protein and growth factors they need for growth, and RPMI inorganic salt solution. And so this allows us to have a medium which is basically the same as our kind of standard T cell culture medium, RPMI. But just with different concentrations of metabolites. So we can take this media formulation, put this on T cells in comparison to RPMI, and just ask, how does changing the metabolite concentrations to make them physiological, how does this change T cell function and behavior? And so we've been working on this project as part of a longstanding, really productive collaboration with Greg Delgoff at the University of Pittsburgh and an extremely talented student in his group, Jeremy Wang. And so in collaboration with Greg's group, we did the following experiment where we took OT1 T cells and activated them with peptide, and then expanded these T cells either in RPMI or for different periods of time in interstitial fluid medium, and then put these cells all back into RPMI and ask what happens when we restimulate these cells by cross-linking. How does their expansion potential change, and how does their functional profile change? And the first thing we found is that as cells spend increasing amount of time in these microenvironmental nutrients, their ability to expand when restimulated decreases. So you can see here, cells in RPMI expand quite robustly, but as cells spend increasing amount of time in interstitial fluid medium, their expansion potential goes down. And in addition to this defect in expansion, the T cells, as they spend more time in interstitial fluid medium, they lose more and more effector functions. They're less able to produce TNF and interferon, as well as granzyme, and they increasingly have higher levels of cell surface PD-1, a marker of exhaustion and dysfunction. And so our kind of key take home is, if we isolate the interstitial kind of microenvironmental nutrient stress, that on its own is sufficient to contribute to the T cell dysfunction. And so is sufficient to cause T cell dysfunction. So we do think that this kind of nutritional abnormalities in the tumor microenvironment are a key contributor to the dysfunctional phenotype that we see of T cells that wind up in these tumors. And we wanted to ask, now using this TIFM platform, can we identify the metabolites that are depleted or enriched that are responsible for these defects that we see in both T cell expansion and T cell function? And so the first thing I'm going to talk about is we looked at metabolites that were depleted in the interstitial fluid medium and asked if those depleted metabolites are contributing to expansion or these function defects that we observed. And so going back to our atlas of interstitial fluid metabolites, we know exactly which nutrients are depleted relative to plasma and RPMI. And so we can take those metabolites and do the same experiment where we have T cells that are expanded in RPMI as our positive control, expanded in TIFM as our negative control. And then we can add back those metabolites that are depleted in TIFM to RPMI concentrations and re-stimulate the cells and ask, if we add those nutrients back, does that improve T cell expansion or T cell function? And so what I'm showing here is when we look at T cell expansion, you can see in the black trace, the cells in RPMI expand quite well. In TIF media for 5 days, they lose their ability to expand. And if we add back in green, one amino acid in particular that's depleted, arginine, that's basically able to rescue the ability of these T cells to expand. And this is consistent with really great work from Roger Geiger, who's shown that arginine is a critical nutrient for T cell growth. And this is depleted in our interstitial fluid medium, and totally consistent with that. If we add the arginine back, these T cells regain their expansion potential. There's also great work from Kostas Iliadis and Marina Pasca-Demagliano's lab that have identified low levels of arginine in PDAC tumors as limiting the number of CD8 effector cells that are in this microenvironment. So we think that we are, this identification of arginine as being limited in the tumor microenvironment is quite relevant to T cell dysfunction in this compartment. Interestingly, we found when we add arginine back, this does rescue T cell expansion, but it doesn't seem to completely rescue T cell function. So if we look at cytokine production when arginine is added back, this isn't rescued to RPMI levels. It's basically the same as what was happening in TIFF media. We do see a partial rescue of granzyme production, but not really a rescue of PD-1 cell surface expression, and a partial rescue of PD-1 surface expression as well. And so this is kind of telling us that the depleted metabolites in interstitial fluid medium, they do limit T cell expansion and to a certain extent their function, but there has to be something else that's going on in this media that's also inhibiting the effector function of T cells. And so what we did was we asked which of the metabolites that are enriched in the interstitial fluid medium, can any of these be affecting T cell effector function upon restimulation? And so again, we can go back to our atlas of which metabolites are enriched in the interstitial fluid, and we can set up the experiment again where we expand T cells, and RPMI as our positive control, interstitial fluid medium as our negative control, or an RPMI where we're adding back these individual metabolites at the concentrations that are in TIFF media, and then re-stimulate these cells and then ask how does their effector function change. And so what I'm showing here is looking at TNF and interferon gamma production in RPMI, or RPMI supplemented with all of these metabolites that are at increased concentration in the microenvironment, or in purple when all of these metabolites have been mixed together. And what we found is that when we mix all of these metabolites that are at high concentration, put this into RPMI, this is able to suppress cytokine production in these T cells. Individually though, lactate, glutamate, glycine, DHAP, which is a central carbon metabolite, none of these metabolites are able to on their own impact cytokine production, but this Kennedy pathway intermediate, which I alluded to before, phosphoethanolamine, has almost the same degree of suppressive ability as the whole mixed pool of these metabolites. And we found on its own, phosphoethanolamine supplementation into RPMI is able to be just as suppressive of cytokine production as interstitial fluid medium. And we also found if we take TIFM, replenish this medium with arginine, and remove the phosphoethanolamine, this is able to lead to about a 50% rescue of cytokine production in T cells upon restimulation. And so we do think that we have identified, well, not all of the metabolites and nutritional conditions in TIFF media that are affecting T cell function. We have found a couple, arginine and phosphoethanolamine, that are particularly important. And we have a lot of information about how arginine affects T cell function, but we know relatively little about how phosphoethanolamine could be regulating T cells. And so we wanted to understand, how does elevated levels of phosphoethanolamine that we're seeing in these tumor microenvironments, how is that affecting and impairing T cell function? And so what is phosphoethanolamine? Over here on the left, I'm showing the Kennedy pathway of which phosphoethanolamine is a component. This is a pathway by which cells can make the membrane lipid phosphatidylethanolamine from ethanolamine as a precursor. And so ethanolamine can be converted by ethanolamine kinase to phosphoethanolamine, CDP ethanolamine before this is with the combination of diacylglycerol used to make phosphatidylethanolamine. And what we found is any of the Kennedy pathway intermediates, when we add these to T cells, they're all able to suppress cytokine production. And so we wanted to know, what is it about really ramping up the Kennedy pathway that is causing impairing T cell function? And so we entertained a number and tested a number of different hypotheses. The one I'm going to talk about now is the one that we think is going on. And this is actually because the Kennedy pathway actually shares some really important metabolic overlap with T cell signaling. So the Kennedy pathway, when it's upregulated, it's consuming a lot of diacylglycerol, which diacylglycerol is a really important component of T cell signaling. And so when T cells, and when the TCR is engaged, T cells produce a lot of diacylglycerol in response to the stimulation. And this diacylglycerol is a really important second messenger that activates protein kinase C, which is critical for inducing the gene transcription programs that are required for gaining activation and effector function. And so what we wanted to know is if phosphoethanolamine in the microenvironment is ramping up the Kennedy pathway in these T cells and limiting diacylglycerol for T cell signaling. And so to test this, what we found is if we look at diacylglycerol levels in T cells by flow cytometry, when we add phosphoethanolamine to cells, this does decrease the amount of diacylglycerol that's available in these T cells. And so we wanted to know, is this reduction in diacylglycerol that's coming from phosphoethanolamine upregulation of the Kennedy pathway, is this what's actually causing effector cell dysfunction? And so to test this, we asked if we have T cells and we culture them in phosphoethanolamine, which I'm showing here on the right, that does indeed suppress their ability to produce cytokines in response to stimulation. If we exogenously activate PKC using PKC activators like PMA, is that sufficient to rescue cytokine production in T cells? And you can see here on the right, in a dose-dependent manner, as we add more PMA and just activate PKC on its own, even in this condition where T cells have less diacylglycerol, this is able to rescue in large part their cytokine production. And so we do think that this is a major pathway by which phosphoethanolamine is blocking T cell function, is by blocking the ability of T cells to produce diacylglycerol in response to stimulation. And so lastly, we wanted to ask, does this phosphoethanolamine inhibition of diacylglycerol signaling, is this something that actually matters for T cell anti-tumor immunity in real tumors where T cells are going to be seeing all kinds of different signals and stimuli? Is phosphoethanolamine, if we modulate that on its own, is that going to be able to affect T cell function and anti-tumor immunity? And so the first experiment we did was we set B16 melanoma tumors in mice and gave them PML T cells to target those tumors, and just looked at diacylglycerol levels in T cells that ended up in the tumor or in lymph nodes. Now what you can see is in the TILs and the tumor infiltrating lymphocytes, there's substantially less diacylglycerol compared to T cells coming from the lymph node. And so that is consistent with a local increase in phosphoethanolamine in these tumor microenvironments being able to suppress diacylglycerol levels and signaling in T cells. And then we wanted to ask if we modulate the tumoral levels of phosphoethanolamine, is that able to change diacylglycerol signaling, T cell effector status, and tumor control? And so to ask this, we generated B16 cells that overexpress this enzyme PCYT2 in the Kennedy pathway, which will soak up phosphoethanolamine by rapidly converting it to CDP ethanolamine. And so this will decrease the tumoral and levels of phosphoethanolamine, and we can then ask what happens to T cells when we inject them that show up in the either control environment or this tumor environment with less phosphoethanolamine. And what we find is when we look at T cells that have that are in these tumors where there's higher PCYT2 levels, they have much higher levels of diacylglycerol. They have higher production of cytokines, and these T cells, these tumors on their own don't have any difference in tumor size or growth, but when they're challenged with T cells, PML T cells, these tumors with less phosphoethanolamine are more responsive and have smaller tumors in response to T cell therapy. And so we really do think that phosphoethanolamine is, even in the context of a tumor where T cells are seeing all these different kinds of signals, this signal, if we modulate the signal, that's able to impact T cell biology and effector status. And so I'll stop there and sum up a few key points from this work. And I think the first thing is I really want to highlight is that the PDAC tumor microenvironment is really an important limitation on T cell on anti-tumor immunity, because it really does drive T cell dysfunction and impairs the ability of these cells to actually kill cancer cells. And then by developing this interstitial fluid medium and making these measurements of what's in the microenvironment, we've been able to identify microenvironmental nutrient stress as really a major driver of that dysfunction that T cells exhibit when they show up in these PDAC tumors. And by building this medium, this has also allowed us to really go in and mechanistically identify which of the nutrients in the microenvironment are driving this dysfunction and how are those nutrients driving this. And so we've identified that there are some metabolites, essential metabolites for T cell biology, that are depleted in tumor environments, namely arginine. And that lack of arginine does indeed contribute to T cell dysfunction, in particular contributes to their lack of ability to expand and proliferate in response to stimulation. But I think really importantly, the other thing that we've identified is that it's not just metabolites that are, the microenvironment isn't just starved of things, but there's also many, many metabolites that are accumulating in this microenvironment. And some of these have really profound suppressive effects on T cells. For example, this phosphoethanolamine, by upregulating the Kennedy pathway, is able to interact with and decrease really essential signaling nodes for TCR engagement and T cell function. So I think that's a really important thing to think about going forward. Is the microenvironment is not just things that have been decreased in decreased in the microenvironment, but there's many metabolites that are accumulating that can have really important impacts on immune cell biology. And we're as we look at different tumors, if we look compared to plasma at phosphoethanolamine concentrations, in many different types of tumors, from melanoma to pancreas tumors to breast cancers, we're seeing profoundly increased levels of phosphoethanolamine kind of across many different tumor types. And so they're starting to really think about phosphoethanolamine as a potentially pretty potent oncometabolite that's immunosuppressive, that could be contributing to the inability of the anti-cancer immunity cycle to control tumors. And so we're very excited to start to look for ways to target and potentially improve the ability of T cells to function when they're seeing this immunosuppressive metabolite. And so I'll stop there and just want to say a big thank you to everyone in our group. Here we are at beautiful Lake Michigan, and in particular Chufan Kai, a former graduate student in our group who did all of this phosphoethanolamine and T cell work, again in very close collaboration with Greg Delgoff and Jeremy Wang at University of Pittsburgh. And a big thank you to our collaborators at the Whitehead Institute, who helped us develop all of our metabolomics pipeline and our funding sources. And I will stop there, and I'm happy to take any questions, and all the questions I'll respond to by email. So very much looking forward to your thoughts.